Antibodies are essential components of the immune system, combating infection and sickness to keep the body safe. Antibody sequencing is a process of determining the amino acid sequence of an antibody or protein. This is served to help identify the antibody structure, and allows large-scale recombinant antibody production to produce the antibody in demand, with batch-to-batch quality consistency. It guarantees a high degree of accuracy, and enables sequencing without having the DNA sequence in advance. Antibodies contaminated or conjugated can also be sequenced, and the results obtained through this antibody production and sequencing mechanism are usually rather precise. At Biomatik, every antibody sequencing project consists of full-length heavy and light-chain antibody sequencing for all species, isotypes, and allotypes. Our de novo antibody sequencing platform works specifically with IG antibodies that do not require access to the cell line, and guarantees 100% coverage of the desired regions and accuracy. Why do you need antibody sequencing? Safety. Hybridoma cell lines are easily lost due to gene loss, mutation, or contamination. De novo antibody sequencing can avoid these problems. Patent applications. To get the protection of your unique antibody through patent applications, sequence information is one of the first steps. Antibody engineering. A 100% sure sequence provides valuable data for further engineering modifications, such as antibody humanization, bispecific antibodies, and antibody-drug conjugates. Recombinant antibody expression. Diagnostic or therapeutic antibody development always requires a stable cell line for producing a high amount of stable antibodies without contaminants. Accurate sequences ensure batch-to-batch consistency throughout research, development, and manufacturing programs. Biomatik provides more than 95% success rate for human, mouse and rat egg antibody sequencing. Standard deliverables include antibody sequencing data and 1 mg recombinant antibody with antibody affinity guarantee by ELISA. We guarantee antibody sequencing results and antibody binding affinity by ELISA for the produced recombinant antibody. Otherwise, it is free of charge. Our fast turnaround time is from 6 to 8 weeks, including antibody sequencing, recombinant antibody expression, and purification, ELISA binding detection. Scale-up capacity of up to 100 grams for recombinant proteins. A one-stop shop for custom gene, peptide, protein, and antibody services. Biomatik has been trusted among top scientists and researchers worldwide for quality products and custom services since 2002. To date, Biomatik has delivered 72,000 plus custom-made products to our customers worldwide. With 200 micrograms of antibody, we can identify all amino acids and deliver the sequencing data within as little as 2 to 3 weeks. With the antibody sequencing data, we can then produce your recombinant antibody with guaranteed antibody affinity by ELISA.